My name is Ameur. I work at the National Genomics Infrastructure at Science for Life Laboratory in Uppsala, Sweden. So what we do is, we get projects from our customers, and then we need to decide on what technology to choose for a specific project. So what we do is often to have a discussion with the person, and then we sort of see what is the biological question that they're interested in. And for example, something like the ion AmpliSeq panel, we're using it quite a lot in cases where people want to look at gene expression counts. And that's a normal thing to do when you're looking at RNA, because then we perhaps don't need more advanced things, like with total RNA that you can get all sorts of information. But using this method, then you can get very good and specific information about gene expression counts. Well, we can do RNA analysis in many different ways. So one way is to do total RNA sequencing, when we sequence all the RNA in a cell, but we also quite a lot use the targeted panels to look and quantify gene expression. Yeah, so the ion AmpliSeq transcriptome, we use it quite a lot for different types of projects that we get. So we are a core facility taking on many different types of projects. But we are using it in situations where we perhaps are using limited RNA and when we're not interested in getting all the information about the RNAs in a cell, but mainly to get a quantification of the RNA levels. So I would say the AmpliSeq RNA workflow has several advantages. So one is that it works for perhaps the samples that are not the highest quality, and also it has a very easy way to get the analyses. So basically we can start a run and then the next day we have a table with gene expression level counts. So it's an easy way to get the bioinformatics analysis sorted. Yeah, so we were very happy to sort of see that a new panel came out for the mouse, because previously we've been using the human AmpliSeq panel, but with the mouse panel we now can do the same workflow also for other types of samples in perhaps experiments where people are using mouse for, it could be many different types of assays where we sort of just are interested to quantify the gene expression counts. So we can now use the same workflow also for those types of samples. The AmpliSeq mouse workflow is basically the same as for the human, which makes it very easy for us. So we can just take this panel and integrate it into our existing workflows. Yeah, so I think circular RNAs is a very interesting class of molecules that have been recently discovered. So they form a circle in the cell, which is kind of a new discovery. And they are also thought to have a biological sort of meaning, but it's still a very new thing that is being explored. The good thing with the AmpliSeq panel is that we can now use a panel. So we actually designed a panel for studying these circular RNAs because they are existing in a quite low abundance in the cells. So we designed primer pairs in the opposite direction. In that way we can quantify only the circular RNAs but not the other RNAs in the cell. So that's a very efficient way to get quantification of those molecules.